So this is a presentation builds very much on what Raphael just showed you for MHC binding prediction, namely T-cell processing and immunogenicity predictions. So briefly doing another biological overview, what does actually happen if you have a pathogen such as a virus infecting a host, as this quite crude drawing of a mouse is supposed to symbolize. You have the virus going inside of the host, entering cells and replicating and spreading throughout the host. So inside a given cell, which is here labeled as an antigen presenting cell, which is pretty much every cell, every nucleated cell, as Raphael was pointing out, the virus is also, as it replicates, subject to degradation into peptide pieces. And these peptide pieces can bind to MHC molecules, which are then presented on the surface. So you end up with peptides on the outside of the surface that represent what is currently being produced inside the cell. And those peptides can be scanned by T cells that have a T cell receptor and a co-receptor. CD8 binds to MHC class 1, CD4 to MHC class 2. And then if they recognize a peptide here that is non-self, that triggers an immune response, and then that peptide is called an epitope. So that immune response could be killing of the cells that presents this non-self peptide. It could be release of cytokines, interferon gamma, the different interleukins, and it triggers for proliferation of the T cell, which can form a memory pool that in the future makes the same response much faster. So when we talk about T-cell epitope mapping, what we really want to know is, given a set of protein sequences that might be representing the proteome of the virus, where are the peptide sequences that can be recognized by T cells triggering this kind of immune response? That's what we call an epitope. So to really understand that, we have to understand this whole process. So that means we have to look inside of this antigen presenting cell of what's actually going on. So, and again, this means what's happening is that you have proteins that are degraded through the proteasome into peptide fragments. These can be further degraded, cut into single amino acids that are used to resynthesize new proteins. So it's a continuous turnover. But some of these peptides can bind to the Tab transporter, where they are brought into the endoplasmatic reticulum, and where they can be further cleaved or bind MHC1 molecules, which protects them from further degradation, and they can be transported onto the cell surface. So you end up then here with these peptide fragments derived from a source protein that goes through these steps. And when you now ask which peptide fragment is going to be out here, that depends on each of these steps, and that is then going to be scanned by a T cell receptor. And each of these steps is somewhat selective. That leads to, from input protein to ultimately recognized peptide. And we have quantified this in the past, estimating that about 15% of all possible peptides make it through this processing. Only 2.5% make it to MHC binding. So this is why we focus on MHC binding a lot, because it's the most selective step. And about 1 in 2 peptides that make it up onto the surface can trigger an immune response based on having a T-cell repertoire available. These are obviously rough estimates, but they give you an idea of the different importance of these steps. So MHC1 is what we're focusing on for MHC binding predictions, precisely because it is the most selective one. Now, these other steps, again, are selective too, and in the IEDB, we have under T-cell epitope processing predictions, these different tools available, specifically this combined predictor that goes through the individual steps that happen. So it combines predictions for the proteasomal cleavage, the Tab transport, and then combines them with MHC binding. So these are trained here on in vitro data sets that evaluated these individual properties. So in vitro digests of proteins by the proteasome, and in vitro transport assays in which different peptides were tested for the ability to be transported by Tab. So the way you use this is you have for these factor, the proteasomal cleavage prediction, you get a choice between immuno and constitutive proteasome. These are two forms of the proteasome, and in a case of an in presence of inflammation, you have typically immunoproteasomes being created, so that would be the default choice. Then for essentially what these do is they look for upstream and downstream from the cleavage side, from each possible cleavage side, how good it fits with the cleavage motif of the proteasome. So you could choose constitutive, but we typically just leave it. And for TAP transport, the lower here, essentially the efficiency of a peptide to be transported by TAP is evaluated. But given that, as I showed you, you have further trimming in the ER, we're evaluating not just the peptide itself, but also potential other precursors. So the overall efficiency of TAP transport is estimated with these two parameters that are essentially have been optimized, and this is what we would, on average, keep them like this and keep them unchanged. So when you combine these, now you can put your protein sequence in. You use your mouse, your MHC allele, just like for MHC binding prediction, and now you have these two additional factors. Can click submit, and then you get scores back, including now this processing score and the total score that combines the processing score with the MHC score. Here, of note, we have inverted the MHC IC50 value so that a high score is high efficiency, just because the proteasome and TAP score go in the same direction. So the combined predictor then gives this output, which essentially the total score is just the sum of the individual scores. And again, as I said, the MHC binding score, and I clicked at the wrong one here. This is the IC50 raw, and then this the MHC score here used in the sum is the negative log 10 of the IC50 value. So that, again, that the higher value then gives a is a better higher affinity. The variance in the scores, like if you look at the different variances in the scores, meaning that these are quite close together, all the proteasome scores and the TAP scores versus the MHC scores fluctuate quite a lot. And that reflects that the MHC binding is so much more selective than the others. So this is how this works. And I think the biggest advantage here of using these tools is that the stepwise process of identifying this is how the proteasome agrees with this peptide, this is how TAP agrees with this peptide, this is how MHC, it gives you an overall understanding where in the processing pathway a given peptide might have an advantage or not compared to others. It has to be pointed out that because of co-evolution of the MHC molecules, they tend to bind peptides that are efficiently generated by proteasome and TAP. So most MHC molecules actually prefer a hydrophobic residue or a positively charged residue at the C terminus, and that's also the signals that are very efficient for proteasome and TAP transport. So if you want to understand these, they're useful to use, and if you need to further limit the set of peptides you want to test, this can be an efficient way using this total score instead of MHC score by itself. But the advantage is rather minor. I think the biggest advantage here is, again, like the understanding of what the different factors are. Instead, what we recommend at this point is to use prediction methods that are directly trained not on MHC binding data, where you have peptides measured in isolation for binding to MHC molecules, but MHC ligand elution data, where you have peptides that are eluted from an antigen-presenting cell, and then these are sequenced essentially by mass spectrometry, and you get large data sets from there. So essentially in this data set, you know that the peptides that you can elude are not only MHC binders, but also they have gone through this processing pathway. So by training a method on this, such as NetMHC Pan EL, you're incorporating essentially the knowledge of what is an efficient sequence for processing and TAP transport or so. And in reality, this just works better, presumably because of the much larger data set than these in vitro data sets for these individual steps. So we recommend using this, and it's a straightforward replacement of the MHC binding prediction methods. It's actually our recommended choice there anyway, again, driven by the large scale of data that is available for training. So this is one of the newer recommendations. Now our recent evaluations have really shown that this EL scores are working better than the combined binding predictions. Another innovation has come specifically actually from the cancer field, is the realization that the expression level of a given antigen that you're looking for is clearly involved in the likelihood of peptides being processed and presented from that protein. So what we have here is, like, if you do RNA-seq, say, of proteins in a cell, and then you have like expression values in terms of TPM, that's transcripts per million, that's the standard output, and look for given different IC50 ranges. You have here as numbers the likelihood of a peptide in this range to be a ligand. So if you're extremely high affinity and you're highly, it doesn't actually matter what expression level you have. But if you're in an immediate affinity range, so like 150 to 700, you see that the ones that have poor expression are rarely contain peptides that are detected by that are MHC ligands. Versus if you have very high expression levels, then that can compensate for the not perfect affinity. So you have this balance of the two, and now we have this as a brand new tool in the IEDB, or not that brand new, but brand new enough that I actually forgot to make better slides for this. I have just this one, but it works like the other ones. The main new thing here is that you now specify, okay, this is a peptide that I'm looking for binding to HLA A0101, and then you add the TPM value for it. So this transcript per million values. And so this works well. And there's this iScience paper from February, where Zainab led that effort, where this is explained in more detail. The main issue is, to some extent, like, where you're going to get your expression value from. So as I said, in the cancer field, this is actually a common thing, that you'll have a tumor sample and evaluate expression levels of proteins, or in general, if you have a cell protein that you're interested in, there's typically easy access to RNA-seq data set. And we actually have a tie-in here to get data from the Cancer Genome Atlas. But if you have something like an infectious agent in a specific cell type, it might be much more challenging to get this data. So then obviously this is not applicable. So that finishes kind of the processing predictions. Here, the immunogenicity predictions now deal with the part of the peptide being recognized by a T-cell receptor. And so there we have done an analysis of looking for data sets in which peptides with similar peptide binding affinity were tested if they were recognized or not recognized by T-cells. And recognized, that means it's an immunogenic peptide, or not recognized, it's not immunogenic. And when we did that, and we looked at the distribution of amino acids in different positions of the peptides, that we found that there was an enrichment of certain residues like tryptophan, phenylalanine, isoleucine, and the depletion of others, including serine, methionine, lysine. So we can take this data and make enrichment scores that are essentially for a given peptide asking how much is it more or less frequent in immunogenic peptides, and use these in this tool, immunogenicity tool, where then the different positions in the peptide are being scored, leaving out the anchor positions, which are first, second, and C-terminal residue. When you run this, you're getting, that's the masking I just mentioned. You click submit, and then you're getting your scores back for your different peptides. This was only evaluated for 9 mers, but we expect that it would also work for other lengths. A high score is more immunogenic, more likely to be immunogenic. And in terms of validation, kind of this is what we get. So 0 would be a neutral score. Above 0 would be associated with being more likely to be immunogenic, and below 0 is not. And you can see that in this data set, like the epitopes that we had were falling primarily, or most of them were at above 0 score, but there's plenty of non-epitopes that also have an above 0 score, but then there's also a bunch that have a below 0 score. So our recommendation is to use this value here as an additional cutoff. You will lose some epitopes though. So as in general, as always, these are additional steps that you can take to make your predictions more stringent. So to summarize here, the processing predictions, or this tabbed and proteasomal buff, give you an insight into the different steps of the antigen processing pathway. We generally recommend using the EL scores as the most efficient way of identifying processed and presented ligands, and they are a straightforward replacement of MHC binding scores. I showed you briefly that we are now incorporating expression values, if available, via AXEL-F, that are then combined essentially with MHC binding predictions, or actually the net MHC EL scores with the expression levels, and that gives an overall improvement in the ability to identify ligands. And then you can further filter them with these immunogenicity scores. So for class 2, Raphael already showed this, and I realize I'm a little low on time. You have this a different pathway. No proteasome, you have exogenous protein uptake, trimming in the lysosome, and they're binding to class 2, and then processing and presentation. So it's a different pathway here. And what we have in this in the MHC2 and P tool is we have looked for the cleavage motif again around the processed and presented class 2 ligands. Essentially, we can use these to predict, given a protein sequence, where are the likely cleavage sites, and that actually improves quite well to identify which ligands are processed and presented on the T cell. Now, interestingly and frustratingly, T cell epitope predictions with this do not improve. And I'll talk about this later. I think I do. But briefly, the reason is that for class 2, you can extend your peptide or shorten your peptide as long as you don't touch the 9-mer core. It'll not impact immunogenicity. So having the exact cleavage site present in the ligand that you are testing for immunogenicity doesn't actually impact its immunogenicity, which was a mystery for us for a while. And but yeah, we and others have shown that now. So there's an example for the tool. You run your predictions, you get your scores back that are here looking for the N-terminal and C-terminal motif. It's like this is 3 residues, yeah, and the cleavage here happens before the P, and here it's between the K and the A, and then you're getting your scores back, and you get an overall cleavage probability for the peptide given the combination of motifs that you're looking for. So, and again, like this is showing we're combining then this cleavage probability score with the binding score. So binding would be evaluating the different cores available between your 2 peptides of interest, and then you'd be getting our overall performance here. And based on that, we chose an alpha value of 0.5, which just combines these 2 in a linear fashion. And that's what we've been using. And here's the paper for it if you want to see more details. And there was a paper also by Morton's group recently that found essentially the exact same thing, that we can boost class 2 natural ligand predictions, but not epitopes, by analyzing these cleavage patterns. So finally, the immunogenicity predictions. There we've done something similar like we tried for class 1, and tried to actually train here a neural network on peptides that were immunogenic versus others that were not at similar MHC binding affinity. And we can combine these score then with MHC binding affinity predictions. And I just said these things. So here's the immunogenicity prediction for class 2. You enter your protein, it runs then the binding prediction method, and then in addition, so it can either do the 7 allele binding prediction method or the immunogenicity prediction, or combines them. And when you run that, you get, you should be very familiar with these kinds of graphs right now, tables right now. You get your 7 alleles back, your median rank, and then the immunogenicity score, which can be combined. and that combined score then is what you would rank by. So what you see here, we have the similar kind of alpha analysis, and it's actually quite minuscule improvements in this neural network. And the other thing with the neural network is always you don't really know exactly what's happening. So it seems to be picking up some signal here, but we're not getting much better than the MHC binding prediction by itself. And so summarizing here, class 2, very briefly, similar as for class 1, we have an enhancement of epitope prediction efficacy that is relatively minor compared to the binding predictions alone. For class 2 also, we have EL scores available now, and we fully expect that those are going to work well as well. We haven't yet performed strict benchmarkings of that. So we would stick to the net MHC2 pan predictions or the 7 allele method if you don't know your specific allele of interest. And I do have to stress this specifically for class 2, we're constantly reevaluating what our recommendations are, and they're subject to change as we are finding out what works better or worse. So I see we have a lot of questions, and I stop here.
We do. Thank you so much, Bjoern, for that. We do have a ton, so I will invite Raphael back. And Raphael, would you like to take the first question in the Q&A?
Absolutely, thank you. So the first question I have here is, do nuclear proteins undergo processing and presentation? That is, are they exported to the cytoplasm and undergo proteasomal processing? Yes, both cytosolic and nuclear proteins undergo processing and presentation. So, Bjoern, are you taking the next one, or should I?
Okay, I just started reading. Can you just take a few?
Absolutely. So how big is the random peptide pool? Yeah, I said a bunch. The peptide pool is 10,000 peptides per size. So, oh, the whole thing is scrolling now. Sorry, go back to the beginning. Okay. Apologies, I missed this detail, but how do you see the IC50? When I run the MHC1 prediction, I only see score and percentile, but not IC50. That's because you ran a method that only gives you a score and percentile and not IC50. One of the methods that doesn't give you IC50 is the IEDB recommended method, net MHC pan EL. If you want to get the IC50, you have to run a method that does that. So net MHC cons gives you IC50. There are others that give you IC50, but that's the one I remember because that was the one I used.
Okay, I can take the next one or another one. Okay, Mario Ostrovsky asks, does a net MHC pan EL 4.1 take into account processing score, or do we do them separately to analyze it? So the net MHC EL 4.1 is trained on the MHC ligand data. So by definition, it's essentially it has in its evaluation of what is a good ligand or not included. So there's no separate processing score, but essentially the output, that score output, includes that itself. Let me do the next one. An anonymous attendee asks, how do you obtain binding affinity binding data from elution mass spec data? Is this a high throughput method? So you do not get binding affinity data back. You get like a present absent value essentially from the mass spec data. And then that's, again, like the reason why we don't have IC50 values as an output, but it's essentially like a probability related score that comes out that this peptide may be presented yes or no. Raphael?
So when a peptide is presented by MHC, does the peptide flanking residues, the peptide flanking residues get cleaved, or is it bulging out on the side or of the binding group? Yes. So in the context of the T-cell immunity, if you remove the peptide, the binding core flanking residues, you'd think they do nothing because they're just flanking there. But if you remove them, you lose stability. So they do interact with the TCR, not directly with the binding group, so it's probably not involved in activation, but it does increase stability. They have to be there.
Okay, I can take the next one. Anonymous attendee asks, can we use our own RNA-seq data with AXEL-F? That's an easy one. Yes, absolutely.
Okay, when narrowing down your list of predicted class 2 binders, would it be problematic to merge overlapping peptides into a longer peptide itself? If I understand this correctly, it's not a problem because the way the methods work is running a sliding window. So you are really doing overlaps of one amino acid at a time. So you can think of it as it takes your sequence and splices in, you take the peptide size and you splice in overlapping peptides. So what you'll be doing is, like, instead of submitting the peptides, submitting an entire sequence. And that's, if I understood the question correctly, it's perfectly fine.
Okay, next question I have is from Mauricio. Can protein levels provided by proteomics analysis be incorporated in the predictions? My expectation is that protein levels will be better than RNA. So we have actually experimented with that. It definitely is better than nothing. One of the problems is how to re-normalize them. I mean, very often, we have to kind of get them on a similar scale like TPM values, but that's obviously possible. To use them directly in, say, AXEL-F. One of the things that is actually quite interesting is, so say if you're talking about class 2 presentation of exogenous particles, and you have, like, say, proteomics data on a virus, that actually does work really well. But if you're talking about MHC class 1, you have a number of peptides, proteins in that virus that are made and immediately degraded again. So you actually don't have very high proteomics data for it. So what we saw is that proteomics data works better for class 2 than for class 1, and RNA-seq data works better for class 1 than for class 2. But the problem there is also, like, for all the class 2 ligand dilution data that we have, it's often unclear what the source of antigen is. So it might be even better than what we've seen so far.
Okay, so for the I'm skipping one here. Can you please clarify the difference between the adjusted percentile rank and the normal percentile rank? So the adjusted percentile rank is the percentile rank adjusted for the frequency of peptide lengths. So that's it.
Okay, so I have a question from Jay Reddy. What is the turnaround time to receive feedback through the help support? Any possibility to speak to scientists over the phone, since the user can get better clarification, easier to communicate? Do you have a mechanism to give a case or reference number to follow through this help support in the future? So our goal is to have within 48 hours for first response, but that first response might just be, in the best case, it's going to be the solution to your problem. But if it's something complicated, it will require that somebody is going to look over it, and it might take a long time to resolve that. Yes, there is a case number, a ticket is open, and that's being referred to, and discussions can go back and forth on it. We do not do phone support live unless you're a licensee, just because there's too many questions. And we're trying to have actually a like an open forum where these things can be discussed. So because we get the same questions repeatedly, and ideally then obviously we want people to benefit from something like Stack Overflow or so, where you can first read what other people have been doing for a given problem. Raphael?
Okay, so for the MHC class 2 recommended method, you said the best method varies based on what alleles you're looking at. Did I say that? I don't know what I said, but there are two ways. So the question is, where do I find this information? There are two ways in which I'm interpreting this. The recommended method for MHC class 2 is a consensus method. So he runs 3 different methods and checks for the consensus. So different methods are going to perform differently. So one is going to be better than the other. But if you're talking about the overall assessment that we do on the methods, which is the assessment that we do to then recommend to you that you use a certain method, then you're talking about the IEDB benchmark. And I'm going to leave here a link for both MHC class 1 and MHC class 2 benchmark, which is what we do. We run a benchmark to decide which method is going to be the recommended one, which is a method that has to be consistently the best one, the best method for a while. So we don't want to change all the time to make you to get you confused.
Okay, I'll take the next one. If we have TCR sequences, are there tools to predict what peptide MHC specific they bind? This is either the next or this is coming up in 3 or so talks. At least our attempt so far, and that is definitely a strong area of research, and it's definitely not trivial.
Okay, so building on the question before about where do I get the results for the best method, there are 2 questions here that I'm going to merge into one. Why are different prediction algorithms better at predicting different alleles? Mostly because they're trained using different alleles, but also because the algorithm itself is different. So they capture things differently. They when you program them, they value different interactions. So with respect to the prediction methods for HLA-1 binding affinity, the question is, how are different tools evaluated and how is the suggestion made? So again, I'm going to leave here 3 links, one for the MHC1 binding benchmark, for MHC2, and I'm going to leave here a link for a paper that we published last year that evaluates our benchmark. So very briefly, what we do is every time a new data set is published in the literature and the IEDB curators incorporate this data set to the IEDB, before any other methods incorporates this data set into their training set, we run the predictors using this data set. So we now have an unbiased assessment of how they're performing. And over time, this accumulates, and now we can make a suggestion of which one is performing better.
Okay, I'm taking the next question from Adrian Noavulano. Why does TePi run only with 50 MERS for MHC class 2? Yeah, so I think that's from our internal bias that we are primarily thinking about applying TePi to scanning protein sequences and getting then optimal peptide sets back, for which we have found 50 MERS to really work the best. They're a great compromise for the longer the peptides, typically the more hits you're going to get, and it's also like a peptide length you can synthesize easy. Now, that being said, I think there's definitely use cases where you want to have a 13 MER or 17 MER for some reason, and that is actually something we should be adding in. So there's nothing terribly wrong with using another size, but again, like 15 MERS tends to be the best compromise. That's why we focused on that. But yeah, we should be adding that as another option.
Bjoern, you can take another one because I'm a little lost here. Which ones should I answer?
Oh, just go through. That's what I'm doing. And yeah, no, if you don't have one, okay. So I'll also, how are we doing on time, Nina?
We have 1 more minute.
Okay, so then why don't I take this one? Peter Chen. It's clear that the percentile method will compare the input peptides with 10,000 random peptides. I wanted to ask, how big is the pool of these random peptides from? Well, that's UniProt. And so that's, I don't know, somewhere in the, I don't know, are we reaching 1 billion yet? I don't know. It's huge.